So in today's video, we will be discussing about the biopotentials, how these biopotentials are generated and how they are responsible for recording a certain activity which is happening inside a certain system. Okay, so let's move to that point, that is bioelectric potentials. As we know, the human body, it is a complex engineering marvel and it has various systems present inside it, and that can be also related to the engineering terms or the technical terms. So different systems like cardiovascular, respiratory system, nervous system, digestive system, etc. All these different systems, they are incorporated inside a human body. So whenever the human body it is functioning, or the systems inside the human body they are functioning, they generate certain monitoring signals, and the signals they provide useful information about their functioning. These bioelectric potentials are actually the ionic voltages that are produced as a result of electrochemical activity of certain cell. So transducers, they are the device which are used for converting this ionic voltages into electrical signals while we are recording the certain activity of a organ present inside the body. So now we will see how this bioelectric potentials they are generated, and before proceeding to that topic we will see how is the structure of cell. So we know that a cell inside the body there are number of cells, and these cells they are the basic building block of human body or any of the living being. Human body has trillions of cells, and these cells they are responsible to take the nutrients from the food that we eat. It convert it into the nutrients, then these nutrients are converted into energy and provided to all the body parts to carry the function properly. So you can see in the figure the structure of cell is shown. So basically a cell, a human cell, it consists of a cell wall, a cell membrane, cytoplasm and a nucleus. You can see in the figure every cell it is enclosed inside a membrane, inside a cell membrane. It maintains the integrity of cell and it controls the passage of certain material inside the cell. You can see over here this blue part, it is the nucleus. This nucleus it contains the DNA and it is surrounded by a nuclear membrane. So this nucleus it also has a membrane surrounding it. Nucleus it determines how the cell will function. This nucleus, you can see over here, it is surrounded by a gel-like fluid called as cytoplasm. It is the medium for chemical reaction. Basically, cytoplasm, that the fluid is responsible for all the electrochemical activities. All the functions of cell expansion, the growth of cell, replication of cell, they are carried out by the cytoplasm. Now we will see how the potentials are generated by the cell while they carry out the function in the further slides. So now the certain types of cells within the body such as nerve cells, muscles, they are encased in a semi-permeable membrane. So what is semi-permeability? Semi-permeability, it is the property of the membrane which allows certain substances to pass while it blocks the certain substances outside the cell. And it is you can see in the figure this membrane it is surrounding your cell body. This fluids, the cytoplasm, the fluid which is responsible for the electrochemical activity, is a conductive solution containing the charged atom known as ions. So we will see the concept called as resting potentials. Now the cell, the cytoplasm, it consists of three principal ions: sodium, potassium and chloride. The membrane of an excitable cell, it permits the entry of potassium and chloride ions, but it blocks the entry of sodium ions. So positively charged ions, they are outside the cell, while the negatively charged chloride ions, they are inside the cell. So inside of the cell is more negative than the outside of the cell, and this membrane potential is called as resting potential. And this potential is always measured from inside of the cell with respect to the body fluids. So resting potential is always negative. We can see over here in the figure If this is the inside of the cell, this is the membrane, and this is the outside of the cell. As you know, the chloride ions, they are allowed to pass, while the sodium ions, which are positively charged, they are outside the cell. So the inside of the cell has become more negative than the outside of the cell. And if we measure the potential from inside with respect to outside of the cell, we get the potential which is negative in sign. And this potential is called as resting potential, and it ranges from minus 60 millivolts to minus 100 millivolt. And the cell in the resting state is called as polarized state. So whenever the cell is in the steady state, or whenever it is resting, then that potential, if it is measured, it is called as resting potential, and the cell in the resting state is called as a polarized cell. Now what happens if the cell is excited? Now when the cell is excited, the membrane changes its characteristics. And now what happens, those positively charged sodium ions which were outside the cell, now they start rushing inside the cell, and the potassium ions which were inside of the cell, they now try to go out from the cell. The sodium ions, they rush into the cell. At the same time, potassium ions, they are now trying to move from inside, but they are unable to move as rapidly as the sodium ions. So the mobility of sodium ions, it is very higher. So as a result, what happens, the inside of the cell, it becomes more positively charged, and outside of the cell becomes negatively charged. Okay, so the potential, when the cell is excited, that potential is termed as action potential, and it is also measured from inside with respect to outside, and its typical range is about 20 millivolt. And this action potential, it is always positive in sign. And the cell, when it goes from a polarized state to this action potential, then it is called as depolarization of cell. So the cell in the excited state is called as a depolarized cell. And the next term comes as repolarization. When the equilibrium is reached, what happens? Again the cell changes its characteristics, and it goes back to its previous characteristics at the resting state. You can see over here, cell comes from depolarized state to polarized state, and that term is called as repolarization. Again the inside of the cell, it becomes negatively charged, as the chloride ions, they are present inside the cell, and outside of the cell is positively charged, as the sodium ions, they are outside the cell which are positively charged. So when the cell again it comes to its original state, that is called as a repolarization. So in this way, wherever the cell is excited, the action potential is generated, and whenever the cell is at the resting position, the resting potential is generated, and this helps in recording the certain activities which are happening inside an organ. So thank you.